go. Well, good morning, everyone. My name is Dr. Terry Flott, and I'm the Secretary of the ASGCT this year, and I just want to welcome you all to this Outstanding Achievement Award lecture and present, and also the presentation of the Senor Scarlato's Public Service Award. I want to welcome so many people for attending our conference. Just to briefly update you, I just heard that we're up to 6,183 registrants for this meeting. So thank you all for that attendance. I'm honored to present two awards this morning. The first is the Senor Scarlato's Public Service Award. It was established in 2003 and named for its inaugural co-recipient, who was herself a tireless gene therapy advocate. The ASGCT's Senor Scarlato's Public Service Award recognizes a person who has consistently fostered and enhanced the field of gene and cell therapy through governmental agencies, public policy groups, public education, and other non-governmental charitable organizations. For 2020, I am thrilled to present this award to Dr. Peter Marks. Dr. Marks is the director of the Center for Biologics Evaluation and Research, or CBER, at the US FDA. In that role, he oversees the development of biological products, including vaccines, blood products, cell therapies, and gene therapies. CBER provides oversight throughout the product life cycle, and under Peter's direction, CBER staff are responsible for reviewing and providing advice during product development, evaluating applications, and making approval decisions based on safety and effectiveness data, and then monitoring of the safety of biological products and conducting research that supports product development and characterization. As the director of CBER, Dr. Marks has created an approach and an environment that has made possible the first gene therapies in the United States. His attributes of flexibility, patient advocacy, and scientific groundbreaking have been the hallmark of Dr. Marks's administration. He has seen through the sometimes arcane details of the regulatory landscape to see the ultimate shared mission that we all have to help the patients. In this way, Dr. Marks has played a key role as the chief enabler, facilitator, and champion of the most historically significant era in our field, the phase of the initial FDA approvals. To have such an intelligent and articulate advocate in the role of CBER director has likely contributed greatly to the progress that the field has seen. Dr. Marks will not be given it this morning, but we do want to sincerely thank him for his service to the gene and cell therapy field and wish him our congratulations. Now to transition to the Outstanding Achievement Award. Now I am honored to present the ASGCT Outstanding Achievement Award. The award is the Society's highest honor, and it recognizes an ASGCT member who has achieved a pioneering research success, specific high impact accomplishment, or a lifetime of significant contributions to the field of gene and cell therapy. It is my distinct privilege now to present this award to my colleague and friend, Dr. Mavis Agbandje-McKenna. I can think of no one more deserving of this award than Dr. Agbandje-McKenna, whose fundamental contributions to the study of adeno-associated virus biology and AAV vectorology have been broad and profound. Dr. Agbandje-McKenna is a highly distinguished structural biologist. Her pioneering work on resolving the crystal structures of parvoviruses led to the opening of an entirely new approach to structure-based vector capsid design. She has also led the new wave of structural biology by harnessing cryo-EM to enable rapid turnaround of capsid structural analysis. Having sub-nanometer resolution on such a high throughput basis has revolutionized the field of vector design. Mavis is the unquestioned leader of this field. The enormous impact of our AAV vectors is witnessed by the fact that there have been 2 AAV gene therapy products licensed by FDA in the US, Luxturna and Zolgensma, and one was previously approved by the EMA in Europe, which is Glybera. The rapid expansion of the AAV field has been enabled by the key role of AAV capsid in enhancing virus tropism for specific tissues. Multiple different approaches to creating new capsids have been employed, including the studies of natural biodiversity, in vitro evolution, and rational capsid design. The approach of evaluating new capsids was mostly empiric in the past. Mavis's work transforms those studies to make them much more mechanistic. She's been able to define the functional significance of many different structural features of AAV and other parvovirus capsids. These structure-function relationships touch on receptor binding, internalization, stability, and immunogenicity. The knowledge generated from her work has had a major impact on the field. Examples of her most impactful publications include her 2008 paper in PNAS on the structure-function interactions of mutating specific tyrosine residues, enabling successful AAV transduction with much lower vector doses. That paper alone has been cited 500 times. Her series of papers in the journal Virology have also been among the most impactful for those of us watching AAV vectorology closely, as she mapped out very specific amino acids required for binding of different receptors. She has quite literally given us the roadmap to the AAV capsid. The full impact of Dr. Agbandje-McKenna's work is just now being felt as different serotypes of AAV advance through the clinic and meet with both success and obstacles that must be overcome with the approaches that are fully informed by the basic virologic mechanisms of how AAV vectors interact with host cells and with our immune system. The field would be much worse off without her unique efforts. In introducing Mavis, I am reminded of the 2001 Academy Award-winning movie, A Beautiful Mind, about the mathematician John Nash, who was gifted to be able to visualize problems in 3 dimensions in a way that others could not. Unfortunately, Nash was also schizophrenic. Mavis shares Nash's ability to see things structurally in 3 dimensions that others cannot. But in contrast with Nash, she is even more remarkable for her collegiality. She is quite simply the most collaborative scientist I have met, and quite exceptionally in our field, is universally well liked by her collaborators and colleagues. Without taking more of her time, let me introduce to you a truly beautiful mind, Dr. Mavis Agbandje-McKenna. Dr. McKenna has a brief pre-recorded acceptance, and then her husband and colleague, Dr. Rob McKenna, will be giving a pre-recorded presentation of the lecture.
It is definitely an honor to be the recipient of this year's Outstanding Achievement Award for the ASGCT. I want to thank the selection committee for their confidence in selecting me. I want to thank those people who wrote the letters of nomination and support. Thank you very much. I want to start by saying that the work that's led up to this achievement is that of many, many people, including collaborators and students, and I thank all of you. And I also want to give a special shout out to my former graduate student, Lauren Druin, who actually got me started in coming to this conference. As most of you know, I was diagnosed with ALS 7 years ago, and thus I get tired now. And so my husband, Robert McKenna, my partner in crime and in the lab, has generously offered to give this talk for me. So I'm going to turn over the presentation to him. Thank you for listening to him. I appreciate your time.
Good morning, good afternoon, or good evening, wherever you are in the world today. As Mavis said, I'll be presenting her presentation today, the title of which is Structure-Guided AAV Vectorology. And if you have any questions that Mavis is unable to answer during the course of this presentation, please just email her at mckenna@ufl.edu, and I'm sure she'll be happy to answer any issues you have with the presentation. So first of all, a few disclosures on Mavis's behalf. She is the co-founder of Stride Bio, which is a company involved in the design of AAV vectors. She is also a scientific advisor board member for Voyager Therapeutics and AGTC. She is also a consultant for the company Intima Biosciences and, of course, Stride Bio. And she has various sponsored research agreements with quite a few biotech companies as listed here. And finally, she's the inventor of several AAV patents that have been licensed again to various pharmaceutical companies. So before Mavis starts to talk, she would obviously want to thank all the people that have been her collaborators in studying AAV. And this list here is individuals, the PIs of labs that have been involved heavily with Mavis on work with AAV. She wants to draw careful attention to certain individuals, the first one being Ken Burns, who was the Dean of the College of Medicine that recruited Mavis to the University of Florida, and obviously had a strong historical vested interest in AAV and wanted to bring a structural component to these studies. Then Mavis wants to acknowledge Nick, who was a basic scientist at the University of Florida, and he pretty much took Mavis under his wing and showed her the ropes and helped her attain her first R01 research grant. And Nick has been Mavis's collaborator throughout her time at the University of Florida. And then finally, she would like to draw attention to Jude and Arun, who were scientists who pretty much introduced Mavis into taking her basic science research and putting it into a clinical environment, which has sort of pushed forward the ideas of using AAVs as biologics. So this is Mavis's group. This is not the full group in any way. Listed here are names of students and postdocs that have worked with Mavis over the last 20 years on AAV-related research. And then on the other side in orange is her current group members that obviously, again, work on AAV. She also wants to thank many technicians that have gone through the lab and the 80+ undergrads that have also been involved in research over these 20 years. She would like to acknowledge also the national facilities, the X-ray synchrotron resources, and also the cryo-EM resources listed here. And of course, she wouldn't be able to do research without the help of all money and these funding agencies listed below, or the grants listed below. This picture is of the 20th anniversary of some of the McKenna Lab reunion group. And I should say that quite a lot of these people, in fact, aren't even related to doing AAV work, because the lab also does a lot of protein work as well. But today we're going to focus in on AAV studies. Finally, Mavis would like to dedicate this talk to Michael Rossmann, who was a postdoctoral advisor. And Michael pretty much trained Mavis and I in viral crystallography and everything to do with research. And Mavis especially wants to acknowledge Michael for giving her the enthusiasm to study science and just stick with it when the problems are hard and just enjoy the pure excitement and joy of getting research done. And without Michael, I don't think we would have ever pushed so hard to achieve what we've done. And so again, Mavis would like to dedicate this to Michael, who sadly passed away last year. So Mavis worked on single-stranded DNA viruses, and these are non-envelope viruses. So there's no lipids involved. These are purely protein capsids. And the T=1 means that they are made up of 60 copies of a capsid protein. Now, over her time, she has worked on Microviridae and Circumventing viruses, and these include and these are circular, single-stranded circular DNA viruses. And a couple of examples is this Spiroplasmin virus 4 here, and the porcine circovirus 2 structure here. She also works on Parvoviridae, which, of course, you all know are the single-stranded linear DNA viruses, and AAV2 is a classic example of those viruses. And finally, she also works on plant viruses, the Gemini-Viridae, and these are very unusual viruses in that they've adapted this T=1 capsid into two incomplete T=1 capsids that can assemble into this geminate capsid. And so these are missing a pentamer of the icosahedron T=1 capsid, but there's two of them. So they're made up of 110 capsid proteins. And the protein, the virus that she works on currently in this area is maize streak virus. And here's the structure of that. So in 2016, the Protein Database did a review on virus structures, and they announced that there were 200 structures put into the Protein Database. And this is just showing the spectrum of structures of viruses that across labs across the world have been achieved. And as I said, Mavis works on the T=1 capsids. So these are these red capsids. These are these small viruses. And these really, really tiny ones, these are the plant satellite viruses. But Mavis works on these groups of viruses here. And on this time of this review, it was that Mavis's lab was responsible for 10% of all the deposited virus structures, be it crystal or cryo-electron microscopy structures, into the protein database. So all these structures are publicly accessible for all to view. So the AAVs belong to the Parvovirinae family, and this is divided into 3 subfamilies, and they package, as I said, single-stranded DNA genomes of size between 4 and 6 kilobases, and the AAV genome is roughly about 4.7 kilobases, and they belong to the subfamily Parvovirinae, so this subfamily here, and in the genus Dependoviruses. So Mavis studies all of the Parvovirinae families, and her group works on pretty much 5 of these genera at the moment and have determined structures in them. And these include diverse viruses as Aleutian mink disease virus, bovine parvovirus, AAV2 that we'll get to in a minute, B19, the human virus responsible for Fifth's disease in children, and minute virus of mice. And one of the important things of all these viruses is that obviously one of their hosts is humans. I should acknowledge here, and Mavis would want me to, is that the Michael Chapman's group was the first group to solve a structure of an adeno-associated virus. That was AAV2. But since then, several groups obviously has recapitulated that data. So AAV now is a biologic, and there's numerous clinical studies going on and trials of using these viruses as biologics. And we all know that Glibera was the first approved treatment by the European Commission, but it was withdrawn about 5 years later. And it wasn't because it wasn't effective, it's because the business model system, it just wasn't cost effective as a treatment. But since then, there have been 2 more FDA-approved drugs. And here we have Luxturna, which was used for subretinal injection. And then also we have Zolgensma here, which is also FDA-approved last year. And this is used by intravenous infusion. So these are just the ones FDA-approved, but there are many more already in line to try and get FDA approval. So I think the movement ahead for AAVs to be used as biologics in gene therapy is very promising. So when Mavis started this research many, many years ago, 20 years ago, first of all, she sat down with a piece of paper and thought, what were the challenges in trying to use AAV as a gene delivery machine? And she wrote down this list. The first one would be obviously the issue of gene targeting specificity. These viruses would need to be able to be directed towards certain cell tissue types or organ types. And so most of the vectors at the time were are still, in fact, trafficked through the liver for clearance. So she wanted to try and understand how the specificities to certain cell types. Another area that she was interested in was the gene transfer efficiency. So even if the virus gets to the right cell that you want it to infect or get the payload into, there are problems with the cytoplasmic clearance and prior to getting into the nucleus. So she wanted to look into how they could make the gene transfer more efficient. Also, she wanted to understand the packaging capacity. As I've already mentioned, the genome delivery of an AAV is around 5 kilobases, which is small for some of the monogenic diseases that AAV would like to sort of touch. So she wants to work on trying to understand how we could increase the packaging capacity. And another issue that she was concerned about, although at the time the literature didn't show it as a sign 20 years ago, was the prevalence of anti-AAV antibodies in a person's blood plasma that obviously would have adverse effects to the idea of using AAV as a treatment therapy. And something that's become more apparent recently is this last issue of production and manufacturing. As AAV moves away from the lab bench and into patients, one has to worry about the vector yield of making more novel capsids and variants and production line, because obviously these have to be cost effective if they are going to be true biologics. With that said, Mavis wanted to familiarize herself with the AAV life cycle to understand how she could use structure to understand these problems. And I'm not going to go into this in detail because it's quite a busy slide, but obviously, first of all, the AAV has to find the cell. It has to then bind to some sort of receptor, be taken into the cell, and then through endosomal trafficking, it has to go and get to the nucleus. And obviously, we'll talk about this later on. There's a pH transition during this trafficking in the endosome. The virus has to be released, most likely through its PLA2 activity that gets activated at low pH, and then somehow it crosses into the nucleus. Not sure if it goes through the nuclear pore or some other mechanism, but it gets into the nucleus, and then we obviously get transcription and the formation of messenger RNA that gets taken out of the cell, where proteins, the viral proteins, can be expressed. They come back in to assemble with the genome packaging, and then this virus escapes ready for reinfection. So this was the typical life cycle of a wild type AAV. When it's outside the cell, obviously antibodies are always around, both the adaptive and also the innate immune response as well. Also could be detrimental, maybe sometimes actually enhance virus infectivity. So Mavis thought that these sort of areas she could start studying. And on the next few slides, you're going to see some of these numbers associated to the heading of the topic or the slide, and you'll see that she's addressing some components of this. I won't have time to go through all of these components today. So Mavis, being a structural biologist, first of all said, okay, you have all these different AAVs, and the literature says they have different tropisms for different organelles within organs within the body. And so her first goal was, well, we need to get lots of structures of these capsids so I can build up some sort of three-dimensional library and understand or get a picture of the diversity of the structures of these viruses. So she set about getting the structures of AAV1 through 9, and then onto the non-human primate AAV vectors, and then even onto variants and even non-primate AAVs as well. The goal was to obtain these structures, and then once you have all these structures, start annotating those structures with respect to things like receptor attachment, trafficking determinants, genome packaging factors, and then what determines where their antigenic sites are on these capsids. And then she could reuse all this information on a mapped atlas of the capsid and use it to engineer second and third generation vectors with improved clinical efficiency. So here's just a list, and looks like wallpaper, but pretty much these are the structures of AAVs that Mavis has accomplished over the years. And here we have AAV1 through to AAV13. We also have the RH10s and some other RH viruses here, just showing you these structures. And to the untrained eye, they will look very, very similar. But if you look very, very closely, say at the edges, you'll see that some have larger projections than others. They're more blunted, and these are things that are happening on the threefold axis. And just to show you, so they have a sequence homology of up to 99%. That would be between AA1 and AAV6. They only have a handful of amino acid differences. And then when you look at AAV5, that's probably about 50 or just over 50% sequence identity to any of the other AAVs. On top of these viruses, Mavis has gone sort of more inquisitive or exploratory in the structures, and she's starting now to look at bovine AAV, bearded dragon AAV, and snake AAV. And I'll just draw your attention to this one, bat AAV. And I think in recent times, we know what can happen when a virus transforms, a coronavirus, a bat coronavirus, going into the human population. We have no immune system available to combat it. But Mavis is starting to think that these AAVs, we would have no immune response to them, as in that we would have never have seen them, but these may be perfect vehicles to be able to transfer genes into human patients. And so this is why she's exploring these capsids now and their structures. And I just want to point out, when you see in parentheses this number here, this 4-letter code number, this is actually a PDB accession number. So you can go to the protein database and you can just type in this 4-letter code and you can bring up these structures. And all the ones with parentheses are ones that have been deposited. Obviously, Mavis still and her team have to deposit some more of these structures into the database. On top of that, as they've developed new AAV variants and chimeras, they've also solved those structures to learn more about the architecture of these viruses. And on the top row is just showing you a list of some of those variants, when some of these are single amino acid variants, and some of them are where there have been loop exchanges as well. And these names will mean people to the collaborators that work on them. Mavis would like to express that most of these structures now are determined by cryo-electron microscopy. And I just want to point out this one at the end here. You see that number at the top there, the 1.86 Angstrom. This is a cryo-EM reconstruction structure, and this is the highest resolution AAV structure today. And it just shows you how cryo-electron microscopy is going to really move this whole field forward, giving structure information at really, really high resolution. And on the bottom here is another set of AAV chimeras, and this time where there have been loop insertions and deletions. And again, I'm not going to go through the integral details of these, but I'll just show you this last one here. Even the untrained eye, hopefully you can see a lot of red here, there's a lot of protein mass at these threefold axes. This variant has a lot of those loops truncated, it's a deleted form, yet it still forms an intact capsid, which sort of is giving Mavis information of what is critical in assembly of a capsid and where you can explore building out on the surface of the capsid to get different phenotypes of these AAV chimerics structures. Okay, so let's have a quick zoom in on the AAV structure. So this is this 60mer of these capsid proteins, and the AAV field will know that this is made up of 3 proteins, VP1, 2, and 3, and most of the capsid protein is made up of VP3. About 50 of those 60 capsid proteins are VP3, and then 5 or so of VP1 and 5 or so of VP2. So they assemble into this capsid. In a structural point of view, the bit that we see is the part that is similar between all 3 of these, the VP3 component. So we pretty much see the ordered structure of VP3 when we show these structures. And this is just showing you a ribbon diagram of the trapezoid structure of the capsid protein. And just showing you some of the beta sheet here in the interior. There's a conserved alpha helix, and then all these loops between the strands are what forms the topology on the surface of the virus. So here's the icosahedral symmetry. We have a 5-fold here, a 3-fold, and a 2-fold. Around the 5-fold, we have a slight protrusion with a pore going into the interior of the capsid. Around the 3-fold, we have these large protruding 3-fold peaks. And then at the 2-fold, we have a depression. So we have like a little valley at the 2-fold axis. And then I just want to point out one other area here that's between the 2-fold and the 5-fold. And we call this the 2-5-fold wall. We're not that original in our nomenclature here, but it may make it easier for you to understand where these components are on the capsid surface. So with all these structures determined, what we could do is a structural superposition of the C alpha coordinates of AAV1 through AAV9 here, and they're all color coded, shown on the bottom here of the slide. And what you see is that the beta barrel, the interior of the capsid, is conserved, and this is conserved in plant and bacterial phages as well. The beta barrel is pretty much an essential part for a viral protein to form these capsid structures. But what you see the variation is in these loops. And Mavis has labeled these loops variable regions, and they're named based on essentially the C alpha differences of these loops. So you can see that these loops have different topologies, and so these are defined as different variable loops. And they also commonly have amino acid differences as well. And this definition of these variable loops came from when Mavis' first AAV structure, which was AAV4, was determined, and she could compare that with the Chapman structure of AAV2. And she delineated these loops, and pretty much the definition and nomenclature of these loops have been kept since that structure many, many years ago. So when we look at these, how these loops decorate the surface, so these are color-coded, these variable loops here in different colors, and you can see now where these loops make the topology of the surface of the virus. In gray is the conserved, more conserved structure. And then in the colored regions are these variable loops 1 through 9 here. And that's the 5-fold pore showing you there for orientation. So once Mavis had all these structures, she hypothesized that, well, you have all these different AAV stereotypes, and they have very, very specific phenotypes in receptor attachment, transduction efficiency, and also antibody recognition. And these must be facilitated by the variations within these loops. These must be the cause of the effect, is the amino acid changes and conformational differences of these loops. While other factors of phenotype like trafficking, DNA packaging, and assembly maybe are more dictated by conserved regions, the core beta barrel, and the amino acids at the interfaces as that icosahedron capsid is being formed. So with that said, and that sort of intro, now I'm going to tell you some of how she can use structural information to tease out functional information. And the first one is the cellular attachment. And Mavis wants to give a recognition to postdocs and students that have worked on this in our lab over the years. And I'm going to just give a shout out to Hazel, Michael, Tina, Linyar, Ed, and Mario, who have worked mainly. The others have contributed, but these are main contributors to this work. And what Mavis realized was that the virus, first of all, binds to some sort of glycoprotein, and this is the initial attachment. It's like the Velcro. There are more specific receptors for any particular cell type, but this is the point of first attachment, binding to those cells. And it's these carbohydrates which are going to be important in that initial stickiness of the virus to the cell surface. And so she studied various AAVs interacting with sialic acid and heparin sulfate, etc. And here's an example over here of just showing you, actually this is bovine AAV and its interaction with a triazole. Okay, so with that, I'm just going to highlight a couple of examples of this work here. And this is just showing attachment of AAV1 with sialic acid, or at least the terminal residue of sialic acid, the terminal residue of the glycoprotein, sialic acid here, at 3 angstroms. And this was done by that old-fashioned method of crystallography. And she found that this binding site was between that 2 5-fold wall and those 3-fold protrusions in this valley here on the capsid. And this shows you the electron density of the sialic acid and the amino acids identified that were involved with those interactions. And over on the other side of this slide here is showing you bovine parvovirus. And it is now showing you the keto trio's interaction with bovine AAV. And again, what I want to highlight here, this was done by Crow electron microscopy, and it was exactly the same area on the capsid, this area between the 2 5-fold wall and the 3-fold protrusion, where that sugar bound. So these are very, very different AAVs, and these are very, very, very different sugar attachments. Yet it is the same cleft on these capsids, which are using different amino acids and different carbohydrates, but the virus is using the same area topology of its structure to do this initial attachment to the cell. And over the years, she's again, as I say, made an atlas of all these different attachments, heparin sulfate and sialic acid, on the capsids of the various viruses. And what this data allows you to do now is you could program in sialic acid binding or deprogram it, or you could program in heparin sulfate binding or deprogram it, because you know the amino acids are involved in those interactions with those carbohydrates. So this is where you can now sort of understand and actually use this structural information to adapt the properties of your capsid. So with that, and because of time, I'm moving on to some other steps that she has been focused on, and that is the endo and lysosomal pathways. And essentially here, again, a big call out to some of the people involved in these work, or the major people that were involved in this: Honju, Bala, Antoinette, Bridget, Justin, and Joshua. And pretty much the idea here is studying what happens to the virus capsid in those early stages of being entrapped in the endosome as they're undergoing that pH transition from about pH 7.4 down to pH 6 and maybe even lower. And so quickly again, this is this trafficking process. And what Mavis is concerned about here is understanding how that VP1U, that unique part of VP1, gets externalized so it can turn on or activate its PLA2 activity and release the capsid from these endosomes. And so the way that she's tackled this is essentially what she wants to do is determine structures and doing circular dichroism and other biophysical methods and understand what happens to the capsid during these pH transitions. And the concept here, and this is only a model, we don't know if this is true, but the idea is that VP1U is in the interior of the capsid at pH 7.5, and then as the pH decreases, and we know this for sure, that the VP1U area becomes disordered or unfolds, and then as we go back in pH, it can refold. And the concept is that maybe it unfolds to allow it to be externalized on the surface of the capsid to refold and therefore exhibit its PLA2 activity and break down those lipids in the endosome. And to this extent, showing you sort of the dedication of Mavis, she has studied AAV 1, 2, 5, 8, 9, bearded dragon AAV and snake AAV, and determined their structures at 4 different pHs, 7.5, 6, 5.5, and 4, to learn general processes of what's happening to this capsid during these pH transitions. Before I tell you those stories, though, I want to make you very much aware, and this is negative stain images, electronegative stains of AAV2, and showing the effects of pH 7.4 on the capsid all the way down to pH 4. And hopefully what you can see is these capsids are fully intact during these pH transitions. So that means that the virus doesn't completely disassemble during that pH transition. In fact, when you do thermal titrations of stability of these viruses, in fact, we find that the capsids are in general most stable at pH 5.5. That is, in fact, when they're the most stable. And they lack stability at lower or higher pHs. Now, this work can also be done, and again, I haven't got time to present it, but looking at divalent cations effects and other effects of buffers as well. And this has implications not only in the virus trafficking, but also in the formulations of the biologics of AAV capsids and storage of these capsids, and what effects that the pH of storage might be having on these capsids before they're administered to people. To show you that this capsid may be not as static as you think, this is just showing you the surface representation of AAV2 again, and we're going to focus in on this area down here near that 2-5-fold wall, and we're looking at variable loop 3 and variable loop 9. This is just showing you the structure of pH 4 and then pH 7.4, and this is a zoom in on those two loops here, and hopefully you can see that these loops have moved. In fact, this structure isn't as static as we might think. In fact, there are conformational changes of loops that are induced upon the change of pH. And again, the variable loop 9 and 3 are involved in this. And so there's some sort of movement about that twofold axis. And you might say, well, does that have any biological implications? Well, maybe it does. I'll get to that in a moment. I just want to show you also at the 5-fold axis, and this is now showing you snake AAV, just changing up the virus here. It's just showing you the 5-fold pore here at pH 7.4 and at 6. And hopefully you can see at pH 6, this pore gets a lot larger. And the pore gets larger because the residues at the N-terminal, or at least the last ordered residues, become disordered as you transition from pH 7.4 down to pH 4. So you see the electron density here at pH 7.4, and then at pH 5, all of these amino acids have become disordered. They don't have electron density anymore, so we can't determine their exact positional location, so that we consider them disordered. And what this does is that means that the 5-fold axis is opened up. And in fact, as you drop the pH down to 4, these amino acids become ordered again. The electron density over here is just showing you a part of that beta barrel core here, showing you that this is invariant over the pH. And that's why the virus capsid itself doesn't break down, but what you're doing is opening up the 5-fold axis. So here we're seeing, as pH is changing, we're seeing structural perturbations at the 2-fold and also at the 5-fold is opening up. And so we believe this is indicative of some mechanism of how that VP1U becomes externalized. I should say, work not shown here, Mavis has also shown using small angle neutron diffraction work that in fact during these pH transitions, the nucleic acid within the capsid also becomes disordered and detached from the capsid cell during those during the reduction in the pH. So all of this is relying on a mechanism of how the virus capsid is opening up, maybe to allow that VP1U to become externalized, but also in maybe allowing the nucleic acid out of the capsid. And just to show you some of this work, the relevance of it, here's a paper where, in fact, if you cross-link the tyrosines across the 2-fold axis of the capsid, you actually, which again is those residues that I was just telling you about, these loops here, you do cross-linking here, you prevent that motion of opening up, you in fact lose the PLE2 activity. In addition, if you make point mutations of a couple of these residues again in these loops, what you do is have a reduction in the transduction of the virus. So again, these are showing structural perturbations, but also these are having real physical effects on trafficking of the virus. So now moving on to the next component of Mavis studies, which is antibody recognition of the capsids. And again, the shout out to the people involved: Brittany, Yushan, Kenan, Nakia, Antoinette, Mario, Ariana, and Shannon here. And all of these people have worked on mapping out antibody binding sites on various AAV capsids. So we know that the AAV virus is obviously going to be affected by antibody responses. And some of those antibodies can neutralize these capsids. And they could neutralize the capsid while it's still in the plasma, that it can never bind to a receptor, or it could be taken into the virus, and it could in some way prevent the endosomal trafficking of the capsid. So there's multiple places where these antibodies could actually cause neutralization of the virus, not allowing that payload of genome to get to its nucleus. So Mavis wanted again to map where these antibodies bind. Okay, and the reason why she wants to do that, if you look at what's challenging in getting the cohorts of patients to do gene therapy is between 40 and 70% of the human population have been exposed to most of the AAVs that we know. That's why she's looking at these more bizarre bat AAVs and snake AAVs, where the human has less probably been recognized by humans. Anyway, so this is pretty much around that 65% sort of antibody pre-existing immunity, which is where we start hitting that herd immunity. Pretty much most people have seen AAVs already in their life. And so again, as I say, that's going to reduce the cohort of patients that would be acceptable to being put into a clinical trial using these capsids. And so they will either be excluded from these trials, or they're going to have to put onto immunosuppressants while the AAVs are being administered. So Mavis said, well, let's see if we can start making variants that can sort of avoid or escape these antibody recognition, make the virus capsids stealth. And so by doing that, what she did was pretty much an onslaught of lots of structures of different AAVs listed here with different FABs bound to them. And what she was trying to do as she built up this library of footprints of these antibodies on these capsids was mimicking what a polyclonal attack of a capsid would look like in blood plasma. And this is pretty much what Mavis saw, was when you put all that data together, all the different FABs bound to AAV1 or AAV5 or AAV9 here, and the gray is the actual virus capsid, and the colors are all the different antibodies. You can see that pretty much they smother the surface of the capsid. So I remember jokingly to Mavis saying, well, there's no way you're going to evade that. Look at it. The things are covered. But of course, Mavis doesn't get deterred by my comments. And she said, no, I think we can do this because the actual bindings footprint are much smaller. And so what we need to do is just that initial binding contact is what we need to prevent. And then we won't occlude so much of the surface of the capsid. And so this is just showing you, zoom in, oops, sorry about that. Showing you, here, sorry about that, technical difficulty here, showing you some antibody structures with various AAVs. Here's AAV1, AAV5, and AAV9. And this is showing you down here with the AAV1, a low resolution 11 angstrom structure, but still you can identify by modeling what loops are interacting with the FAB. Obviously, if you have high resolution structures like this one here at 3.1 angstrom, you actually can see the actual individual amino acids and the hydrogen bonding network pattern of the virus recognition, or the FAB, I should say, recognition to the capsid protein. And then here is an intermediate resolution of 3.9 of AAV9 here with another antibody. And again, shown in this corner over here, showing you the loop that we would know would be the interaction with that antibody. So with that said, now you can actually try and circumvent this pre-existing immunity. You could either do rational design by site-directed immunogenesis of a few amino acids in a particular loop that you know were being recognized by an antibody, or you could take that loop and do directed evolution, which Mavis's collaborators do, and you can therefore change that loop up without changing the properties of that capsid protein. And here's just showing you one example here of wild-type AAVrh10 here. And as you increase the antibody concentration, obviously you stop the effective transduction. But when you mutated that loop, you can allow the virus, obviously, is stealth and not seen by that particular MAB, monoclonal antibody. And then we could also do site-directed immunogenesis, where we change his AAV1 and we've changed up some loops here. And you see that the wild-type AAV1, this is serum dilutions, mouse serum dilutions, and you see that this is neutralized at a much lower dilution or much higher concentration than these variants of AAV. So by essentially now you've allowed them to be stealth to at least the serum to until the serum is a much higher concentration to get neutralization of these viruses. And it's at least at a 1 log scale, even more than that difference. And this can then also be shown, what about humans and people and serum? Well, here's just showing you that rational design approach with the AAV1 variant here. And this is actually a patient who had actually already been administered AAV1. So they had antibodies against AAV1 and then taking their serum and see if this variant can evade being detected by that serum. And again, as you here dilute the serum of the patient, we see that the wild type virus obviously needs a much longer, much more dilute effect before you see GFP expression, whereas this variant actually is showing expression at probably about 50-fold difference in concentration. So again, this is where now this new variant could be used for a re-administration of AAV1 to that particular patient. And over on this side, we're seeing human serum of patients and just standard AAV1 versus this directed evolved version, CAM130. And again, we see that the cohort of patients here, a lot more of them have never seen the CAM130 or their serum evades that, whereas the AAV1, a lot more of these people have antibodies against AAV1. So this again is where you can make a variant now, which is useful and increasing the number of patients that could be administered the virus because they reach the requirement or concentration of human serum where they still could be used. So if you're interested in this work, Mavis has just recently written a review on this, and this is the reference. And this is actually just all parvoviruses in general. So not just AAVs, but FPV, feline parvovirus, bovine parvovirus, and canine parvovirus, and others as well. And this is obviously the work of other labs as well, collaborating labs with Mavis. And what she's done is identify these footprints. And there are 3 such footprints. There's the group 1 footprint, which occurs at the 5-fold. There is the group 2 footprint, which occurs around the 2-fold, and the group 3 footprint, which occurs on those 3-fold protrusions. So each of these have been mapped out, and pretty much we have a really good atlas of the antigenic binding sites of all these classes of viruses. And again, just drawing your attention to the real key points are the 5-fold axis, the 2 5-fold wall, and the 3-fold protrusion. So last thing I have to say about antibodies, or Mavis wanted to say, is we should be worried about them also in our production, because many labs use affinity ligands interactions to purify their AAVs. And so what she's done is a whole load of structures, again, of various AAVs with these single chain antibodies and finding out where they bind. So where is the recognition site that these affinity columns are using to enable you to purify out your virus? And so now she's located these and mapped them, and I really don't have time to go into it, but here's a little diagram showing you there's some that bind at the 5-fold, some at the 3-fold as well. So if you use these, be aware where they bind. And of course, if you've just made a new AAV capsid variant that had amino acids changed, say at the 5-fold, well, I certainly wouldn't use AAVB to be able to purify, because those amino acids will now be changed and this affinity column won't work for that particular virus. So when you're making new variants of your viruses and you need to think along the production line about the purification of these viruses, be aware that when you change the capsid, you're also changing the interactions in these affinity chromatography purification methods. So take care when you're changing these loops. So Mavis has annotated the whole of this surface. This isn't just Mavis's work. This is other collaborators and other people in the AAV field. But pretty much what Mavis wanted to set out, they've achieved, is on the surface of this capsid, they built an atlas or an annotation of all the functionality, be it genome packaging, receptor binding, help in transduction, the antigenic sites, help in assembly, the assembly of the capsid, and trafficking as well. So there are many, many areas that she's been able to annotate on this capsid. Not only Mavis's lab, but of course other labs that do similar work as well. Just to finish up, I just want to show you a couple of clinical vectors which Mavis has been involved with in some way. And again, big shout out to Jude Smolsky here, who pretty much started Mavis off thinking that she could take this basic science and apply it to a more clinical setting. And here, just showing you in blue is one of the surface loops on AAV2, and purple the surface loop of AAV1. And what Jude wanted to do was use the AAV2 capsid, but put in engineering a loop of AAV1 to give better muscle tropism. And so this is what it's showing. The electron density here is showing Jude Smolsky here, who pretty much started Mavis off thinking that she could take this basic science and apply it to a more clinical setting. And here, just showing you in blue is one of the surface loops on AAV2, and purple the surface loop of AAV1. And what Jude wanted to do was use the AAV2 capsid, but put in engineering a loop of AAV1 to give better muscle tropism. And so this is what it's showing. The electron density here is showing you this variant here, this AAV 2.5, and you can see the ordered loop, and you can see that the topology of that loop follows AAV1, but the rest of the structure is still AAV2. So this was a way to adapt the AAV2 capsid to give it better muscle tropism, more like that of AAV1. And here's another example of a structure of AAV2 7M8, which has increased renal tropism, where again, blue is showing you the AAV2 loop, and then the gray is showing you the integration of AAV1's loop. And again, the electron density shows that this loop is fully integrated into that capsid and gave the capsid better renal tropism. And that has just been recently published if you're interested in that work. And then again, just to show one other area of work, and this is a big shout out to Arun and his tyrosine variants, where what we can do is also improve that trafficking of the virus in the cytoplasm. And the idea here was that those tyrosines or surface tyrosines of AAV2 could be phosphorylated, and that would lead to the degradation of these capsids. But if you change those tyrosines into phenylalanines, now you don't have that hydroxyl group, you can't get the phosphorylation, and so far less capsids are degraded and more get to the nucleus. And this is just showing you a GFP study of just showing you the wild type transduction and then showing you the various variants here. And this Y730F variant, and when you look at it on a plot, this is about 10-fold higher in gene expression, transgene expression over the wild type. So this just shows you where you can chemically change just a few amino acids on the surface and increase your payload getting to the nucleus by 10-fold. And again, this again is a big shout out to Arun, getting Mavis involved in such studies. So with all that said, a few years back, Mavis got inspired by her own work, and these two gentlemen had a lot in inspiring her to do this. And Arvin and Pat sort of contacted Mavis, and they decided they would form a company called Stride Bio, Structure Inspired Design. Yeah, it's quite a cheeky name, I think. Anyway, what they did was they used all the knowledge so far. Anyway, they obviously can move their platform to other areas, but they've so far have used these antigenic footprints of the various capsids, and then using Arvin's technology, they can use vector libraries to change these loops out, but keeping the phenotype of these viruses. They create their libraries, they can accelerate the evolution to make new capsids that, of course, they want to make sure they can purify and make in large quantities, where they can have new lead candidates, which are pretty much stealth viruses that the human population will have a less antigenic response to. So again, Mavis wants to thank Arvin and Pat for bringing sort of the basic science of Arvin and Mavis's work together, and Pat being the business manager, helping them get together and actually make this dream come true for them. So with that said, I just want to summarize this talk. Going back to Mavis's original list, she wanted to understand or try and help the gene targeting specificity, and hopefully I gave you a couple of examples, including the AAV 2.5, which increased the muscle targeting. She also wanted to look at the gene transfer efficiency, and I think the tyrosine mutants that she did with Arun are amazing. And then, of course, they've gone on and looked at other post-translational modifications of serines, tyrosines, and lysines. I think they've sort of exhausted the number of amino acids on the surface where you can get modifications, but they're pretty much building up a portfolio of library of what these amino acids and these changes have an effect of the virus as it moves through that cytoplasm. Also, on packaging capacity, I really haven't mentioned that at all, but Mavis has been looking at the interior of the capsid as well, and the charge distribution on the surface, the positive, negative charge. And this obviously is critical in involvement of packaging that negatively charged DNA into the capsid, and by modifying this charge, one might be able to increase the packaging capacity of these viruses. And she's looking at viruses outside of the AAV field to get clues on how this might be done. Then she wanted to look at the prevalence of AAV antibodies, and again, hopefully I've shown you some work where they've shown they've successfully been able to make variants that escape antibody detection. And then finally, the production and manufacturing. Obviously, you can do all this great science, but you've got to be able to assemble a lot of this. You've got to be able to package, make sure most of your capsids are full. And obviously, cryo-EM has been very useful in determining full and empty ratios of capsids. And on top of that, you've got to purify these things. And again, mapping the affinity ligand binding sites is going to be critical, that you know which affinity column you should be using for the purification of your particular capsid that you've designed. So with that, in summary, AAVs have common receptors, trafficking, and antigenic reasons, despite differing tropisms and receptor recognitions and transduction efficiencies. And so this suggests there are common mechanisms, and also this suggests there are common mechanisms for neutralization of these capsids. So we can use mutagenesis work to actually confirm these functional regions, and we can make escape variants. And I showed you very, very briefly, Mavis has a panel of lots of human serum at the moment going on in her lab, where she's trying to understand how many of the human population actually have antibodies against various AAVs, and actually making escape variants of them. And also, finally, I just want to say that hopefully I've showed you briefly some of the clinical significance of this basic science, the production of AAV 2.5, and then the AAV2 tyrosine to phenylalanine variants, which is a proof that the strategy of all of this basic science work comes together and actually makes new novel capsids that maybe have a role in the clinical field. So with that, I thank you for your attention. I apologize, I'm not Mavis, and I may have got tongue twisted on some of the work, and I may have said a few mistakes. In fact, I caught myself saying a couple. I'll let you discover them anyway, but Mavis will be very happy to take questions, and I hope you have a great day today. Thank you, and goodbye.
Well, thank you, Rob, and thank you very much, Mavis. Mavis has asked me to pass on to the audience that she will be answering all of your questions by email. I did put the email address into the question queue so that you could see that, but also if you're having trouble seeing that, we'll answer those questions with the link to the email address. I also want to share that for those of you who haven't seen it, or if Mavis hadn't seen it, that by far the biggest number of comments we have are hundreds and hundreds of votes of thank yous to Mavis and to Rob. Mavis is recognized by the audience, of course, as a true pioneer. So I'll end it here by saying thank you, Mavis, for everything that you've contributed to our field. Congratulations again, and please accept our best wishes. Good afternoon, everyone.
I need to get that.